everybody. For those of you who I haven't met, my name is Melissa Cronin, and I just defended my PhD in the Ecology and Evolutionary Biology department. But it's really fitting that I'm here now, so many years later, because I actually entered the PhD program at UC Santa Cruz as a grad student employee, basically, for the CSP program. So the program is really near and dear to my heart, and I'm really happy to be here with you all today. And I do want to just thank everyone who's listening, but also thank the speakers. I've learned so much over the past 2 days, so I'm really grateful for your sharing your knowledge. I'm going to tell you about my work, but first I want to introduce you, or hopefully reintroduce you, to this incredible group of species, the oceanic sharks and rays, otherwise known as the elasmobranchs or the pelagic elasmobranchs. We have more than 1,000 species of sharks, rays, and closely related species. And they're one of the oldest and most ecologically diverse vertebrate lineages. So they arose almost 420 million years ago. They have really interesting biological characteristics and are particularly vulnerable life histories, but they're really charismatic. A lot of people care about these species. Beyond just being charismatic, they're important for human communities and for ecosystems. So we know that they're functionally important predators, both top and mesopredators that maintain healthy ecosystems. They have high ecotourism value, about 300 million per year. In some cases, they can be important food and livelihood in, particularly in low-income coastal countries. And they have cultural and spiritual value for many indigenous groups around the world. Despite all of these really important reasons, we know that these species are in global decline. So this is from a paper from just last year, which looked at the global abundance of oceanic elasmobranchs, and it found that their abundance has declined by about 71% of the past 50 years. And they attributed this to a corresponding 18-fold increase in relative fishing pressure. So we know that these species are in decline and that fishing is the primary driver. But not all fishing is the same. We know that about 50% of the global catch of these species is bycatch or accidental incidental capture. Basically, you're fishing for one thing, you catch something else. So it's a significant proportion of the global catch. And one of the major sources of this bycatch comes from the global industrial tuna fishery. If you're not familiar with this, it's a massive industry. I like to call it big tuna with a capital T. And this figure is showing catch or landings over time of the major commercial tuna species since 1950. And you can see we have a 1,000% increase over the past 60 years, mainly to meet demand for canned tuna and for sushi and sashimi products. The main ways that we catch these tunas are through two gear methods. The first is purse seine, basically a huge circular net that cinches in the middle, which catches nearly 70% of the globe's tuna. And the second is long line, sounds exactly as what it is. It's a long line with hooks attached to the end. And overall, these contribute to about $40 billion a year in the tuna industry. Sort of fortunately, these fisheries are managed by just 5 regional fishery management bodies, or TRFMOs. If you hear me refer to that, that acronym, I just mean the management bodies that govern tuna fishing, in our case, over an area about 90% of the world's ocean surface. So these are really large areas, but also centralized management bodies, which is will be important later. And we've known for decades that there is bycatch in these fisheries. Sea turtles, marine mammals, if you're familiar with the dolphin safe marine mammal, or excuse me, the dolphin safe tuna label. But less attention has been paid to elasmobranchs, to sharks and rays. And there are several reasons for that. You know, a lot of this catch occurs on the high seas away from coastal areas, which can be really difficult to access, just far from the shore. In many cases, observer coverage can be low. In some cases, less than 5% of vessels have an observer on board taking data. A lot of particularly the elasmobranch data is confidential. And as a result, we have really not a whole lot of real baseline information about how many individuals these are catching and what's the impact. So the broad question of my thesis was, how do we manage threatened but extremely data poor species? In order to answer that, I broke it down to 4 sort of sub questions, which I'm going to run through right now, but I won't really go into all of them today. The first, I really just wanted to get some of that baseline information. So what is the magnitude and impact of this capture in industrial fisheries. How many are we catching and what is the threat? Secondly, I took a genetic approach. I looked at population structure for a high priority group of elasmobranchs, or the manta and devil rays. I won't talk about that study today just because it's a little bit weedy, but if you are interested in population genetics and mantas or devil rays, I'm happy to discuss that later on. Next, I looked at how can fisher perspectives inform precautionary management for elasmobranchs, particularly for those manta and devil rays. And that's the chapter I'm going to talk about today. And then finally, I looked at tuna fishery policy in those large management bodies and how we can improve it for these species. I'm going to, as I said, focus on the fisher perspectives chapter, this third one here, but I think it's important to give you context. So I'm going to run through a little bit of the findings of this first and third chapter so you sort of understand the broad context of this work. So starting with this, this study. So essentially what I did was I took a subset of pelagic elasmobranchs, 22 species, 13 sharks, and 9 rays. And these species, as you can see here, are all considered of conservation concern, listed on the IUCN Red List and or on CITES, which controls trade. And I'm not going to go too deeply into this, but I'm surely happy to talk about the methods later. But basically, I created a reported catch database by harmonizing catch data from each of those 5 management bodies. I had to convert a lot of that data, clean a lot of that data, and I created this harmonized catch database. But as I told you already, it's really likely that we're not getting the full catch reports of what's actually being caught at sea. So next I had to create an estimated catch database. Basically, what do we think we're actually capturing based on catch rates? So I did this big literature review. I took 344 catch per unit effort rates, basically how likely you are to capture a certain species with a certain gear in a certain region. And I combined that with effort data to derive catchability coefficients. You can use this then to calculate an annual estimated catch, basically a range, a confidence range of an upper and lower estimate. And then I combined those estimates with stock assessments to try to say, okay, what proportion of that catch is overfished or fished sustainably? I'm going to show you some of the results of that work right now. And first, I'm just going to show you a subset of the data and how elasmobranchs stack up next to those charismatic, as I call them, species for which we've had a lot of conservation and research interest over the past few years in this type of fishery. So on the y-axis here, you can see mean annual catch in individuals, and on the x, you can see the different groups. And it should sort of jump out at you that elasmobranchs make up about 97% of the reported megafauna bycatch in these fisheries. This is really important because, as I told you, these species have been overlooked. However, it does make sense. They're more common, more frequent in space and time, more abundant. So there's obvious clear reasons for that. But if we look at just the elasmobranchs, we can see an interesting. Excuse me, I'm going to explain this first. We can see an interesting pattern in the data that I looked at. So again, on the y-axis, we have mean catch in individuals. On the x-axis, I broke this down by a gear type. So long line and purse seine, the 2 main gear types. And what I'm showing you right now is just the reported data in black. I'm going to show you now what we estimate that they catch. So I hope it's very clear in this figure that the estimated catch, basically what we think they're actually catching, really dwarfs the reported catch. In fact, so these fisheries report just 2.9, about 3 million individuals per year, but we estimate that the catch is actually about 7 times higher than that. So clearly we have this underreporting problem, in addition to a lot of individuals caught each year. But if we have a lot of individuals, we don't necessarily know what that means. Does that actually mean that there's a threat? So like I told you, I wanted to look at the proportion of these species that are overfished, overexploited. Here on the y-axis, we have the proportion of the annual catch. And on the x-axis, I have these 3 bins of population status as produced by a stock assessment. Not overexploited, basically anything that is considered sustainable, overexploited, and then unknown. And you can see here that at least 30% of the elasmobranch catch is overexploited according to these stock assessments. But we have a whole nother 10% here in this unknown category that we have no idea the population status for them. And I'll just add that it's more likely that those are overexploited than not, just because they're data poor, potentially rare species. So just to wrap sort of the findings of that first study, I showed you that these fisheries report 3 million individuals per year, but that the true catch is probably a whole lot higher, and that catch is about 30% overexploited at least. But something great about this work is that it involved co-authors from 3 of those tuna commissions in the Eastern Pacific, the Indian Ocean, and the Atlantic Ocean. So we're really excited about how they can use some of the results of this work to actually guide their own research and management priorities. Okay, I'm going to move on to the chapter that really was the focus of my CSP work at least. And I'll just give you sort of a cliff notes of my genetics chapter, which found that for manta and devil rays, there are some really serious genetic flaws or genetic issues that suggest that we need precautionary management for these species. But let me introduce you to them first before we go into that. So there are 9 recognized species of mobulids, manta and devil rays. Together we refer to them as the mobulids. And it's been suggested that they have this dangerous dermography. As I said earlier, this is some sort of gratuitous drone footage that we took in Baja of the monk's devil ray. But all mobulids have slow growth, late maturation, and few offspring, just 1 pup every couple years. And while they can be these sort of smaller animals, about 40 pounds, they can also be very, very large. In fact, the largest species, the oceanic manta ray, can reach the weight equivalent to a small sedan and the width, wingtip to wingtip, that a giraffe is tall. And as a result, these species have been subjected to both historic and current fishing. Now we have very important or very significant fisheries for their meat and for their gill rakers, which are used in some traditional medicine. And as a result, we have these observed population declines all around the world. Any site here in this map in red is where a population decline has been observed. But now we know that one of the most important sources of mortality for these species comes from the global tuna purse seine fishery. Again, that fishery I discussed earlier that cinches, a large net that cinches at the middle. This map shows the distribution of fishing effort for this fishery, which, as you can see, overlaps quite nicely with the distribution of mobulids globally. And the area that I'm going to talk to you about today is the Eastern Tropical Pacific, which is a really important, very large tuna purse seine fishery, and catches about an estimated 3,000 mobulids annually. And I'm going to show you kind of what it looks like when a mobulid is captured on board one of these vessels. Here you can see 2 or 3 crew members releasing a mobulid off the side of the deck of a large-scale industrial vessel. And you can see in this case that the release is really ad hoc. They sort of use a cargo net to just dump it overboard. And in this case, the animal sort of swims away relatively happily. But we don't know yet what proportion of these animals are surviving capture. And at least preliminary evidence suggests that the method of release and handling really matters and can really, in fact, determine whether or not the animal survives. We also have this recent study that showed that post-release mortality is the most important, reducing post-release mortality is the most important conservation action we can do to improve their status. And then, as many of you probably know, we know from decades of research that fisher-informed bycatch mitigation is just more effective. There's also right now a lot of interest from the industry in reducing this mortality, specifically because they want to avoid boycotts and achieve sustainability certification. So it's the best time to act for mobulids right now in this fishery. But what we didn't know was at what point in the fishing operation we can actually intervene. So I'm just going to give you a quick primer on tuna purse seine fishing. There are 3 main stages to the fishing operation. The first is before setting, before you put your net in the water. Secondly is before brailing. Brailing is the process of bringing tuna on board the vessel deck. And the third is during that process of brailing, bringing tuna on board. And so I was interested, when is the first moment that we can intervene to actually reduce this mortality that we want to see? So what I did was I conducted a participatory research project with tuna fishery participants, mostly in Ecuador. I surveyed 170 stakeholders. Those were fishers, skippers, captains, mechanics, observers, basically anyone with experience on board the vessel. And those were fishers from vessels flagged to Ecuador and to Spain. I recorded some demographic data, and I also conducted focus groups with 3 fishers. I'm going to show you some of the results of that. Just one result from that paper. But again, the question is, when are you first aware of a mobilid? In other words, when is the first time that we can intervene? And this is a Likert plot. It's showing you participant responses on a scale of never to always. So answers on the right in green and teal suggest that those fishers always can see the animal in that stage of fishing. The results are really not surprising. You know, these species are much more likely to be sighted later on in the fishing process, which is what we sort of expected. But I was actually sort of surprised that some fishers are aware of mobilid before it even reaches the vessel deck. And that's really promising because it means that we can intervene earlier, which for these species might be important. I'll also add that a couple of the folks who responded that they always sight a mobilid in the water before setting the net were helicopter pilots, which is really interesting and cool. In this fishery, vessels use helicopters to sight schools of dolphins and the tuna that associate with them in this ocean. And so I have now a student, an undergraduate student, who is doing a survey project specifically targeting helicopter pilots. So we'll have a little more information about whether we could harness their knowledge in addition to actually avoid these species in the first place. We also use this work to identify some really relatively minor operational modifications that could help improve survival for these species. And the one that I'll call out here in the bottom right of the screen is this really amazing innovation that was designed without any prompting by scientists or managers by fishers in this fishery. And what it is basically just a grid using materials already available on the vessel deck that can actually act as a sieve or like a pasta colander to catch the animal before it reaches the vessel deck. This was really exciting because it was this sort of novel, simple tool that could be really impactful for these species. So I'm just going to share with you where we're going building off of that design, which is a video of a design produced by some of my collaborators at ASTI, specifically Jefferson Marua. And this is basically a formalized version of that same grid you just saw. And you can see this grid right here. It's a steel grid that sits over the hole in the vessel deck where tuna are dropped to be flash frozen. This is a brailer where you can see a few mobulids being caught alongside the tuna. But as the tuna are released into the vessel hold, this grid, essentially very simple steel grid, will actually catch those mobulids just like a sieve so that they can be released off the vessel deck very quickly. The cool thing about this is that it's really easy to design, it's very simple, and it's really quick. Mobulids, something I haven't mentioned, is that they are obligate ram ventilators, meaning that they must pass water over their gills constantly to stay alive, like many shark species. And so every extra second that they sit on the vessel deck is potentially a likelier mortality event, unlike something like a sea turtle or a dolphin, which breathe air. And so the quicker that we can get them off the vessel deck, the better. And in this case, this video from start to finish is 3 minutes long, which is really a relatively light speed for this type of operation. So you can see them being lifted over here by the hydraulic winch. So where we're going with this work is I'm going to actually go on board the tuna vessel this summer and tag some of the animals that have been released with this method and see if we can detect any measurable differences in survivorship. And it's really exciting work to come. One last thing I'll share from this work, and then I'll wrap up, is we really wanted to bring fishers into the process as well as give back something to bring them into sort of the production of science and conservation. So I worked with an artist to design a poster that helps with species identification, which is very difficult for these species. They look really similar, as well as correct and prohibited handling techniques. We translated this into, I think, 9 languages and made it available both online and in physical form to fishers all over the world. This, you can't really tell, but is the poster on board a 360-ton tuna vessel in the Eastern Pacific. So that's really exciting, and we're growing access to that poster on the regular, which is cool. Okay, that was the bread and butter of my my DE work, but I'll just share one last finding from my this policy paper to kind of contextualize this work in the broader global tuna fishery. And this work's focused on how we can actually improve fishery ecology policy and management for these species, given the fact that my work identified these serious problems for some of these vulnerable species. So I conducted a quantitative and qualitative policy analysis of 4 of these regional fisheries management bodies. I identified 34 policies that pertain to elasmobranchs, and I coded those policies against a rubric using a risk-based mitigation hierarchy framework. What is a risk-based mitigation hierarchy framework, you might ask? Well, you might be familiar with this if you've ever done biodiversity conservation, risk mitigation. It's basically a way of contextualizing the different actions you can do in response to a threat. In this case, it acknowledges that fisheries use different strategies to address bycatch. Those go on a spectrum of things that avoid bycatch to things that really don't have much of a probability of reducing mortality. And I won't go too deeply into them, but very briefly, I took those policies that I identified from the regional management bodies and I fit them into the mitigation hierarchy. Basically, what do they tell us to do for these species? And I'll show you those results very briefly, but I think that it should stand out at you immediately. The overwhelming thing that we found was that only one of these policies contains an avoidance approach, despite the fact that we know that avoidance is the most impactful thing that we can do for elasmobranchs. So this is a clear gap in management and conservation for these species. One sort of good silver lining is that these policies are relatively new overall, and those policies that are stronger, those that do avoidance and minimizing of bycatch, are relatively recent. So maybe we're seeing sort of movement in the right direction. All in all, I think that these policies are opportunities for conservation. I showed you that they're new and increasing, but that they're currently inadequate to avoid capture. But again, they influence this catch over such a huge geographic scale, so they're potentially really impactful. Okay, to wrap up this work, I know that was sort of breakneck speed, but I showed you that these elasmobranchs are caught in substantial numbers and that at least almost a third of that catch is overexploited. I showed you that these fisher perspectives can inform creative management and hopefully be scaled up in the future. And then I showed you that we have a lot of work to be done in these policy realms. As for my future research directions, I'm really fortunate to be headed to the lab of Javier Baserto at Duke this coming late fall to look at the conservation and food security implications of large and small scale fisheries interactions. So happy to talk about that work. I'm sure it overlaps with a lot of the interests of folks in CSP. And with that, I want to thank my partners and funders, particularly ISSF and IATTC, which were the main partners for my designated emphasis work. I'd love to re-thank my committee and advisors, particularly Don, who was my faculty advisor for my designated emphasis, as well as Katie Sito, who has been incredibly helpful through this entire process, as well as all of these new social science skills that I learned. I want to thank the Coastal Science and Policy Program, and particularly Anne and Sarah, who have been so wonderful in letting the DE students into the fold. I've learned so much from being a part of this program, and I'm really, really grateful. I think it totally changed the sort of direction of my research, and I'm just so grateful for that. I want to thank the Fishers again for their constant help and support and sharing their time with me. And I'm happy to take any questions you might have or talk offline. Thank you so much for listening.